Hello, everyone. Thank you for joining us for another of our Mission 3-Day webinar presentations. This is Carrie Stovall. I'm the Director of Events here at Komen at the Dallas office. So we are excited to have everybody with us today. And we're going to go ahead and get started because I know we have this scheduled for 30 minutes and we want to take advantage of all of that time. Just a quick update. I just wanted to say, we're already, it's kind of hard to believe that we're already into the 3-day season. We've had Michigan and Twin Cities events already, and this weekend is the Seattle 3-day. So if there are anybody that's listening that's walking this weekend or crewing this weekend, I just wanted to say good luck. It's going to be a fabulous event, and we're excited to have the Seattle 3-day taking place this weekend. So before we get started with Dr. Pietenpol today, we wanted to kind of give everybody an update on some exciting news that was launched yesterday. You might have seen this in your social feed, over email, or even on your local news stations, as I know some of our local affiliates had coverage there. Is Komen announced yesterday through the media that we are launching what we would call our big bold goal, and the organization's been working on this tirelessly for a while, and we're just really excited, and I'm just thrilled to see all of the energy being put behind this goal. And with our 3-day community, I know the 3-day community doesn't back down from any challenges, so this is not going to be one that's going to back us down. It's just going to make us stronger and keep us going. So I'm just really thrilled. So we're going to start off, and I'm going to turn it over to Michelle from our mission team to give an update on the Big Bold Goal, so you guys can kind of have some more information on it, as well as I know that we as the 3-day will also be working on some materials to get you about this, so we can use this as you go out and talk with your donors and the 3-day community. It'll also start to be incorporated into the on events for the remaining of the year. So Michelle, I'm going to turn it over to you.
Thanks so much, Carrie. I'm Michelle Martin-Pozo. I'm Director of Research Programs here at the Dallas office. And as Carrie said, I'm going to tell each of you a little bit about our new bold goal and mission statement to start off the webinar. So our vision is a world without breast cancer has remained steadfast since the inception of our organization. However, Komen is coming upon our 35th anniversary, and to celebrate this point in our history, where we've been and where we must go, we are refocusing our organization to making this vision a reality. Our new mission statement encapsulates our organization's unique approach to ending breast cancer forever. Komen's mission is to save lives by meeting the most critical needs of our communities and investing in breakthrough research to prevent and cure breast cancer. We believe this mission is more important than ever now because despite tremendous progress against this disease, 40,000 women and men still die of breast cancer every year in the U.S. This is a number we have never been willing to accept, and one that we will change with our new bold goal. Komen's bold goal is to reduce the number of breast cancer deaths in the U.S. by 50%, and to do this all within 1 decade. We are uniquely positioned for this goal, and we can achieve this by drawing on our unparalleled leadership to take on this bold goal on 2 fronts: by funding breakthrough breast cancer research and by working to ensure that every person has access to and receives the highest quality breast cancer care. Our bold goal sets an enhanced focus on finding new treatments for aggressive and incurable forms of breast cancer by leveraging Komen's expertise in identifying and funding the best and most innovative breast cancer research and researchers. We count on our advisors, the world's most influential cancer scientists, clinicians, and advocates, and we will draw on their expertise as we enhance our focus in areas such as finding more effective treatments for metastatic disease, developing better therapies for more aggressive forms of breast cancer that have become resistant to treatment over time, and leveraging the use of transformative next-generation technology that can detect and allow treatment of primary or recurrent breast cancers at their very earliest stages. We know that research is critical to ending breast cancer, but we've also learned over 3.5 decades that all the research in the world won't help us if the benefits can't reach those women and men who need it most. Our bold goal has enhanced our focus to close that gap in breast cancer outcomes within the communities served by Komen. This strategy is threefold. We will continue to identify and address what drives racial and ethnic disparities. We will work to eliminate any barriers to timely use of quality care, which includes screening, diagnostics, and treatment. And finally, we will ensure support throughout the continuum of care through patient navigation. We invite you to learn more about our Big Bold Goal by going to our website to find more details about how we will achieve this in the impact section of our website. In addition, you can learn more about our new fall ad campaign, More Than Pink. And without further delay, please let me formally introduce our speaker. Dr. Jennifer Pietenpol, one of our esteemed Komen Scholars, began her research focused on the signaling of one of the most commonly mutated genes in all of cancer, known as P53. At Vanderbilt University Medical Center, she serves as a director of the Vanderbilt-Ingram Cancer Center and has combined her research expertise in molecular genetics with bioinformatics in order to identify differences in triple-negative breast cancer. Her studies are responsible for the discovery of subtypes in triple-negative breast cancer and has led to several clinical trials aimed to help oncologists determine the most appropriate and precise treatment for those breast cancer patients. Dr. Pietenpol has been recognized many times for her leadership, research, and mentorship to early career investigators. And most recently, following Dr. Pietenpol's presidential appointment to the National Cancer Advisory Board, she was appointed a member of the Blue Ribbon Panel, which advises Vice President Joe Biden's National Moonshot Initiative.
And so before we turn it over to Dr. Pietenpol, I forgot one thing. This is Carrie. When I did the introduction, is just to remind everybody, if you participated in one of the webinars before, that on the right side of your screen in the webinar, you will see a chat area. So during Dr. Pietenpol's presentation, if you have questions, please utilize that format of submitting questions in the chat area so that we can, have those available at the end if we have time for Q&A. And then if we don't have time or there's additional questions, we will work to get those answered after the webinar and get those out to you. So I just forgot to remind everybody about that format, and so we will turn it over to Dr. Pietenpol.
Sorry, I think I was on mute, so I will try to recapture what I just said. I just want to say good morning or good afternoon, depending on your time zone, to all of you who have joined today, and also tell you that as a Komen Scholar, I'm committed to the bold goal. I want to thank all of you for what you are doing to raise funding for research, and I want to share with you today how that funding has impacted our ability to get advances in the area of one particular type of cancer, triple-negative breast cancer. I also want to emphasize how we use the power of basic and translational discovery to address the major challenges that Michelle just talked about. In particular, you're going to hear today how we're using this funding to get insights into metastatic breast cancer, in particular triple-negative breast cancer. And so as all of you are aware, there's been great progress from hard-won research of the past 2 to 3 decades in the area of breast cancer. And approximately 60 to 70% of breast cancers in women we define as estrogen receptor positive. That estrogen receptor has been a target of therapy for those women who have that subtype of breast cancer. For 20 to 25% of women diagnosed with breast cancer, their tumors express another receptor called HER2. That receptor presents another opportunity because we have drugs that target it, and those receptors are known initially to drive breast cancer growth in those women. However, we have a major challenge in that 15 to 20% of breast cancers that are diagnosed are what are called triple-negative breast cancers, and that name defines what they aren't. They are negative for estrogen receptor, they are negative for HER2, and they are negative for progesterone receptor. The challenge is that to date there have been no targeted therapies for women who are diagnosed with triple-negative breast cancer. We know these cancers occur more often in young women. We know they occur more often in the African American population. We also know that many times with high frequency, those women who have germline mutations in BRCA, more likely than not, they tend to be diagnosed with triple-negative breast cancer. And so there's no targeted therapy currently for triple-negative breast cancer, and we're trying to change that. And we are doing it with the help of Komen funding. And that is the story I want to share with you today. So my laboratory is very interested in discovering why triple-negative breast cancer is such a heterogeneous disease or diverse disease, and decipher that diversity so that we can actually get insights into new therapies for women who are diagnosed with it. On the left side of the slide, you will see that there are 8 different triple-negative breast cancers shown by the view of a pathologist through a microscope. And just looking at that picture, you can see the wide variety of phenotypes or appearances that these tumors have. From this view, it is not possible to determine what is the best therapy for those women. We are trying to figure out the molecular basis of these various tumors, discover what's driving them, and then identify what we call biomarkers or predictors of response or clues in the tumor that will help us align women who have these tumors with the right therapy. So what I'm showing you on the next slide is a scientist's view of when we subtype tumors according to the genes they express. So in 2000, the human genome was sequenced, and a lot of genomic technologies came on board. That enabled scientists and investigators to begin to subtype disease based on the wide array of genes that would be expressed in a tumor. So for example, in 2000 and in 2001, investigators in the breast cancer field subtyped breast cancer largely into those subtypes I first talked to you about. ER positive, HER2 positive, and then triple-negative breast cancer. We had great interest in the last 5 to 10 years in taking new cutting-edge genomic technologies and seeing if we could subtype this very difficult to treat and challenging triple-negative breast cancer. What I'm showing you on the right side of the screen is what we call a heat map or a gene expression map. So it may look like a holiday card to some of you with the green and the red, but what you are actually looking at here are 587 triple-negative breast cancers in which we have arranged them or organized them according to the genes they express. And you can begin to see that groups fall out based on the genes they express. And red means they highly express a gene relative to another gene. And so from this type of analysis, we were able to actually gain great insight into triple-negative breast cancers. And in the next few minutes, I want to tell you how we got to these subtypes, these various groupings of triple negatives that have different biology, how we're translating that to patients as quickly as possible in the form of therapeutics, experimental therapy, and what the impact has been. And so we started by leveraging worldwide data. So we collected around the world, this was all available through data sharing, 21 breast cancer data sets from 8 countries, and this represented 3,247 cases of breast cancer. From those cases, we were then able to take molecular tools and informatic tools and identify 587 triple-negative breast cancers that have been collected and analyzed by investigators around the world. So in 2011, when we started this Komen-funded study, we were categorizing triple-negative breast cancer into one category. Women were being given pretty much standard of care, one type of treatment, and if they were enrolled in clinical trials, everyone, it was an all-comer trial in which there was no knowledge that there were many different biologies in these tumors. After our analysis of 587 triple-negative breast cancer tumors, we were able to subtype the disease based on the biology, which we learned from analysis of the genes that were expressed in these 587 tumors. So we were able to now have a pie chart in which we had different biologic subtypes of the disease. Since 2011, again, underwritten by Komen funding, we've been able to analyze another 767 triple-negative breast cancers, and from that now more succinctly subtype the disease into 4 different biologic subtypes. I'm going to tell you a little bit about those in a minute. But then importantly, analyze the entire environment in which that tumor arises and grows and get clues as to the neighborhood or the environment in which the tumor is growing in a given woman. And so now with those 4 subtypes, we also define whether there is an immune reaction or immune cells within that tumor or not. And that's very important because of the advent of immunotherapies and the possibility of aligning some of those immunotherapies, which have been very successful in other tumor types such as melanoma and lung cancer, and seeing if we can use those and investigate their potential in triple-negative breast cancer. So I will come back to that in a few slides, but at this point in 2016, our research has led us to looking at 4 different subtypes of triple-negative and then having descriptors of the environment in which those tumors grow. And so what's one of the first things we did with the data we generated from our studies? The first thing we did was we shared it. We developed a web-based subtyping tool for triple-negative breast cancer in which any investigator worldwide can enter their data and subtype a tumor and learn more about it. We have about 1,000 users a month to this subtyping tool, so about 72, we've had users from 72 countries. Many of the users are pharmaceutical companies, and the hope is that they're using this tool and the ability to subtype the disease to identify new drugs for patients with triple-negative breast cancer. And so the ultimate goal is to really personalize the therapy for women with triple-negative breast cancer, that we could have a molecular profile, understand the genes that were expressed, and overlay that with any mutations in those tumors, and then use that information to identify the right drug for the right patient at the right stage of their disease. And that's really the major goal of these studies. And so let me take you back to the 4 different subtypes and just briefly talk about these and what it means. There are 2 basal-like subtypes. They have what we call epithelial features. So breast cells are typically epithelial. We know from knowing the genes that are expressed how these basal-like subtypes will align with current therapies. I really want to focus my attention today on what we consider the lowest hanging fruit of these studies and findings that we thought we could translate to women in the most rapid fashion possible. And that is, I want to tell you about what we call the luminal androgen receptor subtype of triple-negative breast cancer. This subtype expresses the androgen receptor. This was a big aha, because the androgen receptor has been associated with prostate cancer and as an oncogene driver or a cancer-causing driver of prostate cancer, so unregulated androgen receptor signaling. Further, we noted that women who expressed androgen receptor in their triple-negative breast cancers, about 40 to 50% of them had mutations in another gene. This name is just an acronym, but it's PIK3CA. That gene encodes a protein that drives tumor growth. We knew that pharmaceutical companies around the country were making inhibitors to that particular protein product when it comes to regulated in human cells. And so we began to realize that we now had a subtype of triple-negative that potentially had 2 drivers, 2 ways for the tumor to grow, androgen receptor as well as this mutated PIK3CA gene, and that we had the discoveries which could be the convening power to bring pharmaceutical companies together to try and use drugs in women with triple-negative breast cancer that had not been used before or used in combination. And so in collaboration with the clinical principal investigator of the study, Dr. Vandana Abramson here at Vanderbilt, we were able to rapidly move this to clinic by using the Komen-funded basic discovery as the convening power for 2 major pharmaceutical companies to supply us with a drug that targets the androgen receptor called enzalutamide and a drug that targets the gene product of PIK3CA called tocilizumab. And the first thing we had to do was test the safety of using these 2 drugs in women that have never been used in combination before and determine a maximum tolerated dose. And so we began the phase 1 safety trial here at Vanderbilt. This has all occurred in the last year. That trial accrued 14 women and men from across the United States and from the states shown here in this map, and it had huge impact. So remember, the phase 1 clinical trials to test to determine the maximum tolerated dose of a drug. You're not really testing efficacy at that point. However, the impact of the phase 1 is that the patients enrolled were already having disease stabilization. We're already having amelioration of their symptoms. Shown here are a few patients of Vandana Abramson's. Vandana smiling here, this gentleman here on the left who gave us permission to use his picture, traveled from Alaska for this trial, is a man with breast cancer and had disease progression while on the, disease stabilization and reduction of his symptoms while on the trial. This is tremendous. For these individuals, this was an amazing trial from the standpoint of getting targeted therapy for their disease. So the next phase in that is now we have to move to a phase 2 trial, and we have to test the efficacy of this combination now that we've determined the maximum tolerated dose of the two drugs together. And so we do a randomized 2-arm trial to determine if triple-negative breast cancer patients will benefit from inhibiting the androgen receptor and inhibiting this other protein which we call PIK3CA. And so we're currently enrolling patients from across the United States. They'll either go on to one arm of the trial, which is enzalutamide and tocilizumab, or they'll go on just the AR inhibitor alone, enzalutamide. The key thing to take away from this is that at each stage we are getting biopsy material. So again, translating back to the laboratory, we're able to do further discoveries and identify those key features of these tumors in women and men that may make them more susceptible to this treatment or actually resistant to this treatment. And that's tremendous discovery that we can do to actually accelerate the use of these new targeted agents. So again, I want to stress to you how Komen has really impacted our ability to do this work quickly. So the discoveries were made at a bench in a lab at Vanderbilt University. We've translated them to a phase 1 study, and now the phase 2 study is open throughout the entire Translational Breast Cancer Research Consortium, which is funded in part by the Susan G. Komen Foundation. This consortium includes 17 different cancer centers, and thus this trial can be offered to men and women in these cancer centers who have androgen receptor positive triple-negative breast cancer. And so again, the power of the research funding that Susan G. Komen provides now gets to be disseminated in all of these different areas of the country. And so now I want to return back to this concept of immunotherapy. I just told you about how the research discoveries at the bench are being translated and potentially offering a future benefit for women with triple-negative breast cancer. So that's about 16% of triple-negative breast cancer. What about the others? What are we doing today for those who actually are suffering from metastatic triple-negative breast cancer, and what are we trying to do to find new routes of therapy? So as I said before, we have subtyped to 4 different groups, and also we now have molecular clues and biologic clues about the neighborhood, the tissue context in which that tumor is growing, because we're now analyzing the immune features of these tumors. And I want to direct your attention to the lower right to a graph which we call a bee swarm plot. And it's called a bee swarm plot because each of those little points or periods represents a patient's tumor. What you see in black here are patients who immune descriptor would suggest they do not have an immune reaction or immune cells in the context of that tumor. Those in red are patients' tumors that actually have a number of immune cells infiltrating into that tumor. The tumor has put up some type of shield and probably blocked the activity of those immune cells. And that then gives us promise that for those individuals, we could use some of the newly developed immune therapies to reawaken or recharge that immune system. Importantly, the data analysis we've done would suggest that women who are diagnosed with a triple-negative breast cancer that is a mesenchymal subtype here, you can see that these are in these tumors, there's an absence of an immune descriptor, an absence of immune cells. And for those women, perhaps, the approach of an immune therapy at this point wouldn't be indicated. And in fact, we would want to spare those women any adverse effects that could come with that therapy because their benefit may not be there. And so again, using the convening power of the Translational Breast Cancer Research Consortium with Vandana Abramson as the PI of the trial, we are now entering in and opening up throughout that consortium a phase 2 trial for metastatic triple-negative breast cancer, in which we will start to understand if these new immunotherapies actually work in a high frequency of patients with triple-negative breast cancer, determine what the markers or predictors of our response in men and women, and which patient population to use these agents in going forward. To answer all those questions, we need to do this clinical trial, which again will be a randomized 2-patient to 1 patient on the arms shown on this particular slide. So some patients will get the immune therapy plus a standard of care carboplatin, and other patients will get carboplatin with the option to cross over if they should progress. Again, we will collect tissue biopsies because that allows us the power of discovery again back at the bench to try to understand the molecular features of these tumors that would make a patient sensitive to this treatment or perhaps insensitive or resistant to this treatment. Knowing both of those is very important. And so again, with the Komen funding, which all of you have been highly engaged in securing, we've been able to advance precision oncology for triple-negative breast cancer to the form now of investigational trials. I put back up the first slide showing the pathologist's view of the disease under a microscope, and now I put labels saying these are the subtypes of that disease, which actually gives us insights into some of the molecular features of the disease as well as some of the gene expression and biology of the disease. We are identifying predictive biomarkers or markers of response to standard of care therapy and new agents. We're discovering pathways that are driving the tumor growth. And we're further defining the disease, which means we can become much more personal in our care and personalize that care for each patient. Obviously, none of this would be possible without the support from Komen, without an amazing multidisciplinary team. Many other members of this team are also funded by Komen, including Brian Lehman, who is a lead investigator in these studies, Vandana Abramson, who is the clinical investigator who drives the clinical trials, as well as an amazing group of pathologists, biostatisticians, and obviously trainees as well. I also want to thank all the women and men who are participating in these clinical trials because they are actually allowing us to decipher the different subtypes of triple negative and importantly those proteins and genes which can be targeted in the disease. And finally, I want to thank all of you for giving your time today. All of you 3-day walkers and crew, the race participants, the donors, your friends and family, your continued support is essential for us to accelerate these advances and really for us to achieve that bold goal. I just want to thank you and obviously would welcome questions.
Dr. Pietenpol, this is Carrie, and I think we are pretty close on time, so we are going to do the questions electronically now that we have them collected, and we'll get those answers, work with you on those answers, and then we will get those to the participants of the webinar. So I just want to thank you, Dr. Pietenpol. We really appreciate your time today, and this information I know just energizes me. I've heard it before, but I get energized every time about the progress that we're making, specifically for triple negative. So thank you.
I just want to thank the 3-day community and all of you.
Thank you. So with that, we're going to wrap up because we are on time. So with that, just a few things. I want to make sure and let you know that we did record this session, so we will get that along with the Q&A part posted so that if you were not able or you have team members that weren't able to participate, that you have access to the information that Dr. Pietenpol presented, as well as we will also be sending out a post-webinar survey. So we encourage all of you to participate that and provide us feedback, as this is a series of webinars that we want to continue, and we feel like your feedback will help us improve these or look for topics and make sure that we can continue to get you the information and the impact that your hard work and your dollars are raising. So thank you very much for everybody for joining us, and thank you for all the work on the 3-day, and we will see you next time.
Thank you all.
Ray will want you on.